Welcome back to Biology 102. We finally finished plants, we finished with the creation of Earth, we finished how everything has come about. Now we're going to start our introduction into animals. So for the next couple of units or several units, we're going to be going over animals, animal diversification, and then we're actually going to go into those systems for these animals. So we're starting out with Chapter 18, our introduction into animals. Here we're going to look at our first scientist for this lesson. The scientist, his name is Theodor Schwann. He was a German physician who was born in 1810 and he died in 1882. He's going to attend medical school and study medicine at the University of Bonn and University of Würzburg. And the University of Berlin, This is going to be where he's going to earn his medical degrees in 1834. He's going to continue his work as an assistant to a famed physiologist, Johannes Müller, at the University of Berlin in 1834, and he does this until 1839. Here he's going to perform research on the structure and physiology of cells. Studies are going to result in the isolation of pepsin, which he's going to discover in 1835. He also is going to help to identify the yeast's role in alcohol fermentation in 1836, and he discovers specialized cells called Schwann cells. Now, Schwann cells are actually going to be named after him because he is going to find these cells that are going to start to wrap around the axons, and he notices with these cells that wrap around the axons, they're going to be able to insulate that axon and cause that action potential to travel a lot faster down the axon. So since he discovered it, he is going to be the one that we named this cell after. And so these are going to be named in 1839. And then he's going to also coin the term metabolism in 1839 as well. He's best going to be remembered for his detailed study of cellular compositions of animal tissues and their structural similarities of plant tissues that's going to lead to the cell theory in 1839. Now remember with the cell theory, that's going to go in and say that all cells are made of other cells, and it goes in and says all cells have to come from pre-existing cells is how it's worded, and that genetic information is going to be carried from a parent cell down to the offspring. So that is with your cell theory. He's going to be appointed professor of anatomy at the University of Louvain in 1839 to 1848, and professor department chair of anatomy, physiology, and embryology at the University of Liège in 1848 to 1880. So that kind of takes him towards the end of his life. Remember, he passes away in 1882. He's going to become a member of the American Philosophical Society in 1863. He gets the Copley Medal in 1845. He goes in and becomes part of the Royal Society in 1879. And then finally, he becomes part of the French Academy of Science in 1879. Now going into diversity. The animal kingdom is going to consist of an estimated about 1.5 million described species. These are all going to be multicellular eukaryotic species, which are going to lack cell walls. Just noticed I'm covering up the notes. So these things are going to lack cell walls. And so here we're going to see some different animals. We've got a tiger, we've got jellyfish, earthworms, we have a praying mantis. With these animals, these are going to be heterotrophic animals. Heterotroph means that they're going to be able to consume and ingest other organisms. There's going to be 5 different types. Oh, it looks like I froze. There we go. The first one is going to be carnivory. These are going to be animals that eat other animals. Next, we have herbivory. These are going to be animals that eat only plants. Then we have omnivory. These are going to be animals that eat both plants and animals, and so that's like most of us as humans. There's going to be parasitism. That's where they're going to feed on other living organisms and usually cause harm to that organism. And we have detrivory. This is where an organism is going to feed on dead, decaying organic matter. That matter is called detritus, and the animal eating it is called the detrivore. Now with mobility and symmetry, mobility is going to be found in all animal species. That includes free-swimming larval stages of sponges, which will ensure survival through irritability. Now mobility is movement, so all animals at some point in their life will be able to move, even though sometimes they will settle down and then become sessile. Remember, sessile means that they're stuck to one place and that they can't move. So at any given point or at a certain point in an animal's life, it will have some form of mobility. Now symmetry, there's going to be 3 different symmetries that are going to be based on the use of imaginary medians that are going to divide the body parts into equal halves. Asymmetry is going to lack a definite organization of body parts, and it prevents division into equal halves along that median. If we talk about radial symmetry, this is going to be where you can have numerous median planes that are going to pass through central axes, and they produce equal halves. So think of a pizza, think of a pie, things like that. And then our third one is going to be bilateral symmetry. This is where body parts are going to be divided along a single median plane to produce equal and right halves. So you would cut like kind of down the center of the body. We, for example, as humans, are going to be animals that are going to have bilateral symmetry. Ooh, sorry. That's sitting up here. So with this, that bilateral symmetry is going to be associated with cephalization. What is cephalization? Well, cephal means head, and so that's going to be a concentration of localization of sensory organs such as the brain or things that you would find in the head. So cephalization is going to refer to the head. Terms to describe specific regions of animals are also going to include things such as anterior. That can be consistent of the head region. Or if you're talking about an animal like we are, who stands upright, that is going to be the front portion of the body, so anterior. If you're looking here at this goat, the anterior would be the head of that goat. And so you're going to see around this area here. Sorry. If you're talking about posterior, that's going to be towards the tail end, or if it is an animal that stands upright, it will be towards your back, so that's the posterior region. Dorsal is going to be backside. Ventral is going to be belly side, so that's going to be towards the front. So with us, the anterior would be in the front, and the dorsal would be in the, ventral would be in the front. Posterior in the back, dorsal in the back. Now, if you're looking at this animal here that's going to stand on all fours, it's going to be different. So anterior is towards the head, posterior is towards the back, dorsal is its back, ventral is its belly. So it would be different for one that stands on 4 legs versus 2. So we're going to have 3 steps to embryonic development or fertilization when it comes to these eukaryotic animals. First, we're going to have cleavage. This is going to be where the zygote is going to proceed through a series of rapid mitotic divisions to produce an embryo that's going to be composed of several smaller cells known as blastomeres. 2 types of cleavage that can occur. The first one's going to be a spiral cleavage. This is going to be what produces an 8-celled embryo where blastomeres are going to be arranged within grooves of previous mitotic divisions to exhibit a spiral appearance. If one or more of these cells separates, they result in defective embryos that are going to fail to develop properly during the determinate cleavage. This is going to occur in things like flatworms, also known as planarian, mollusks like snails, and arthropods like insects. Our second type of cleavage is going to be radial cleavage. This is going to result in an 8-celled embryo whose blastomeres are going to be arranged in 2 layers of 4 sitting on top of one another. During development, if one or more of these cells separate, they're going to proceed through what's called an intermediate cleavage, and they're going to develop into a complete embryo. This occurs in animals such as echinoderms, which are going to be your sea stars, and chordates like vertebrates. Next, we have our blastulas. These are going to be developing embryos that become a hollow sphere that's going to be composed of a single cell layer of cells that are going to surround a fluid-filled cavity known as a blastocele. Third, we have gastrulation. This causes the arrangement of the folding of the blastula cells that become the gastrula with 3 distinct germ layers. These are going to be your ectoderm, which is going to form the outermost germ layer, the mesoderm, which is going to form the middle layer, and then the endoderm, which forms the innermost germ layer. These are going to fold inwards during that gastrula stage, and they're going to form an internal body cavity known as the archintron, and that's going to develop into the embryonic gut. So here's going to show the two different types of gastrulation in developing eukaryotic organisms. So first are going to be the protostomes. The protostomes are going to start to develop mouth first. So they develop mouth to anus. The deuterostomes, which you're going to see at the bottom, are going to be the ones that develop anus first or gut first. And so they develop from anus to the mouth. Can you guess which one we are as humans? We're actually deuterostomes, so we develop anus first. And so it starts to develop that opening. Now with this opening, it is actually going to travel through our entire body from mouth to stomach, or anus. And so with us, anus starts to develop first, and it develops up into the mouth. And so that's going to create basically a hollow tube. And within that hollow tube, anything that you take in through food actually stays in that tube. Now it gets broken down, it gets digested, you will absorb nutrients out of it, but anything that's not absorbed into the body stays in this tube that you see developing here and exits out through the anus. And so with that, it's kind of interesting. You can think of yourself as a donut or a Twinkie. Whatever goes in will come out, but it can absorb some of the nutrients and things in from that food that's going to be in this tube. Now, 3 steps following fertilization are going to be, we have this gastrulation here. So the first part that we're going to see here is going to be what we call diploblastic. That's where the gastrula is going to consist of the ectoderm and the endoderm. These germ layers don't develop further. Radial symmetry is going to be common in animals here like jellyfish and sea anemones. Then you can have what's called a triploblastic, and that's where the gastrula is going to contain 3 germ layers, with the mesoderm located between the ectoderm and the endoderm. So it's going to have a continual development of that triploblast, and inside of that, it's going to give rise to important things such as organs and organ systems. These are going to be bilaterally symmetrical animals, so things like flatworms and even humans. The archintron of gastrula is going to open to the outside of the blastosphere and is going to form the embryonic gut of animals and surround the blastocele. Radial symmetrical animals are going to have the archintron develop into a gastrovascular cavity. There you're going to have a 2-way gut where food and waste are going to enter and exit through the same area. Bilaterally symmetrical animals, the archintron is going to become a 1-way gut where the tube's going to complete with a mouth at one end and an anus at the other. Now, with these body cavities, bilaterally symmetrical animals are going to have a blastocele cavity that's going to be filled with mesoderm to form 3 body configurations. You have acolymates. These are going to be things like planaria. These are going to be animals with a mass of mesoderm tissue that's going to fill the entire blastocele. You have these pseudocolmates. These are going to be your roundworms. These animals are going to have a false body cavity that's going to be surrounded by a mesoderm extending along the outer edges of that blastocele and endoderm. Then you have your colmates. These are going to be animals such as humans like us. These animals are going to possess a fluid-filled column, which is going to be completely lined with a mesoderm. So you can see that down at the bottom there, right in the middle. So that's what ours is going to look like. Now with different body plans, bilaterally symmetrical animals are going to be considered protostomes or deuterostomes based on the order of the embryological development of their mouth and anus. Now, like I just told you, we're going to be considered deuterostomes, but there are some animals that will develop mouth first. So animals whose mouths will form prior to the formation of the anus are going to exhibit things such as spiral or determinate cleavage. And these are going to be examples of mollusks or arthropods. Deuterostomes are going to be animals whose anuses will form first prior to the mouth, so anus first. These exhibit radial symmetry or intermediate cleavage, and these can be things like sea stars or even humans. Now going into the different defined tissues. Defined tissues will consist of cells that secrete extracellular matrix. These are going to be intricate, non-living layers that contain water, proteins, and polysaccharides. So if you look at this picture below, it's going to show you a cell membrane. With the cell membrane, you're going to see on the inner portion that you're going to have microfilaments of the cytoskeleton, but on the outer portion, that's where you're going to find your extracellular matrix. This extracellular matrix you will see with these proteins like collagen. Sometimes you'll have elastin there. Sometimes you'll have reticular fibers. Along with these, you'll see that you'll have carbohydrates that are attached to these proteins, and then you'll also have a polysaccharide chain. Familiar components are going to include the proteoglycans, collagen, and elastin fibers that we see here. It's going to help to provide structural integrity to these tissues, so it makes them not only tough, but it also helps them to stretch and to return or recoil back into their shape and to keep their shape. They're going to play an essential role in proper functioning of these tissues, and they're going to help to influence the development of specialized organs and organ systems. Now, going into 4 different types of tissues. First, we have our epithelial tissues. These are consist of sheets of tightly packed cells that are going to cover external surfaces, and then they're going to line the internal organs of the body, organ cavities and different organs. So if you look over to the side, you will see that we have different shapes, and we have what's called simple or stratified. So this is how we're going to characterize these layers. If we say it's simple, that means it's going to consist of one layer of cells. If we say it is stratified, it's going to consist of multiple layers of cells. Now, they're also going to be distinguished according to what shape they are. So first, we're going to say if it's one cell layer thick or if it's multiple layers. So that could be 2 or more. That's stratified. Next, we're going to add in that shape when naming them. So you can have flattened cells. These are going to be referred to as squamous. You can have cube-shaped cells. These are going to be referred to as cuboidal. And you can have column-shaped cells. These are referred to as columnar. These are all going to help to provide protection, and they're going to help to serve other important functions, such as secretion or excretion, or even absorption within the body or even on the outside of the skin. Looking over at the pictures, pencil real quick. So notice that your squamous, these are your flattened cells. Stratified squamous is going to be 2 or more layers. Simple squamous, 1 layer of flattened cells. Cuboidal. Simple cuboidal is going to be 1 layer of cuboidal cells. If it's stratified cuboidal, it simply has to be 2 or more layers. Columnar is going to look like columns, and of course, simple is going to be 1 layer. Stratified is more than 1 layer, so 2 or more. Sometimes you'll see that they're going to have microvilli on top, or they could even have cilia on top of them. Next type, we're going to have connective tissues. These are specialized tissues that are composed of non-cellular matrix, or what we call a ground substance, surrounded by cells and protein fibers such as collagen and elastin. These are going to serve a multitude of functions such as support, protection, binding, storage, and even transportation. 6 primary types of connective tissue are going to be as follows. We first have here the loose connective tissue that you're going to be able to see. This is going to be characterized by a watery ground substance that consists of loosely packed collagen and elastin protein fibers. So you will see the elastin fibers are going to be the skinny purple fibers there, and then the collagen fibers are going to be the thicker fibers. So the collagen is going to help with structure and support. It is going to help to, let's say, keep your face from sagging, things like that. So you start to lose collagen as you get older. The elastin fibers, they allow for stretch and recoil. And so those are important in allowing for structural support. By allowing things to stretch and not stay stretched out or distended, it allows it to also come back. Also in here, you will see fibers that are going to be secreted by, these fibers will be secreted by the cells that you see around them called fibroblasts. So those purple cells are your fibroblasts that are secreting these fibers. So altogether, they're going to function in supporting organs and securely binding the epithelial tissue. Our next type is going to be the dense connective tissue. With dense connective tissue, it's going to be a supportive tissue that's composed of densely packed collagenous fibers and fibroblasts within a watery ground substance. And so those fibroblasts, remember, they're help making those collagen fibers. These are going to be found in places such as tendons, where they have to secure muscle to bone, or ligaments that secure bone to bone. The next type, we're going to have adipose tissue. Adipose tissue is another name for fat tissue. So it's connective tissue with lightly packed adipocytes or fat cells that are going to be relatively few, loosely arranged, with protein fibers embedded in a watery ground substance. It's going to be significant as a storage site for energy. So anytime that you eat more than the amount of calories that your body needs, it's able to store these for use later, and it stores it in the adipocyte or in the fat tissue. This tissue also helps to provide insulation for the body, and it helps to cushion organs. And so any type of adipose tissue that's going to be around an organ is going to be referred to as visceral fat. The fourth type is going to be cartilage. It's going to be composed of a flexible, jelly-like ground substance with a densely packed collagen and elastin fiber structure going through it, along with chondrocytes. Chondrocytes are going to be cartilage cells. These are going to occupy small pockets within that matrix, and those small pockets are referred to as the lacunae, or lacunae if it's multiple, so that's plural. This is going to be common between places like the vertebrae of the backbone and the ends bones, where you're going to have articular cartilage, and it's also going to be found in respiratory tract passageways. So you'll find it in places like the trachea and the bronchi. Our fifth type of connective tissue is going to be bone. Bone tissue is going to contain collagen fibers. These collagen fibers do require you to have plenty of vitamin C in order for them to be produced. And then you're going to have osteocytes. These are going to be your bone cells. So these are found in lacunae as well, just like with the cartilage cells or the chondrocytes. But this time, instead of being surrounded by a jelly-like matrix, they're going to be surrounded by a solid ground substance that's reinforced with mineral salts, such as calcium and phosphorus. And we often refer to that as hydroxyapatite. That's the name of the salts. These are part of the strongest connective tissues in the animal kingdom, and they're going to provide support and protection of other tissues and organs within that animal's body. And the sixth type is going to be blood. It's characterized by its watery matrix, which is referred to as plasma, and comprised of proteins and specialized cells. These include things such as red blood cells, which carry oxygen, white blood cells, which are going to help to defend against infections, and platelets, which are going to promote clotting. If there's any type of damage or injury within the body or outside the body, that clotting is going to be important. It functions in transporting gases and nutrients and other substances around the body, such as hormones. It also transports waste products through the body to areas that can get rid of it. Another type of tissue is going to be muscle tissue. So that's going to be your fourth type of tissue. We just finished with connective, which was your second. Remember, we had 6 types of connective. So now we're going into the third type of tissue, which is going to be muscle tissue. It's composed of cells known as muscle fibers or myocytes. These are going to be able to contract, and they have excitability to them. So muscle fibers, which is another name for a muscle cell, these in skeletal muscle and cardiac muscles are going to be what's referred to as striated. Striated means it looks like it is striped. Smooth muscle will not be striated. So the first two, A and B, that you see at the bottom, these will be striated. So when we look at these underneath the microscope, we're going to look for those striations. Cardiac muscle is going to be stimulated involuntarily. It's controlled by the nervous system, and it causes contractions of the heart. And so really you're going to have a pacemaker that's going to set off those contractions. Skeletal muscle is actually the only muscle that's going to be voluntary, and you can control it, so you can move yourself from place to place without relying on the nervous system to actually go through the processes. You can actually do what you need to do with that. Cardiac, you're not going to control the beat of your heart. Smooth muscle also is going to be involuntary, and these contractions are going to happen in spots like blood vessels and in hollow organs within the body or within the intestine. Our fourth type of tissue is going to be nervous tissue. This is going to be used for forms of communication. They form a network that allows animals to react to stimuli. They're composed of the cells, which are going to be referred to as the neurons. These are responsible for transmitting nerve signals to effectors, such as other muscles or even glands. They're composed of neuroglial cells, which are going to be cells that kind of hold them into place and help them out some and fill in spaces. Neuro means nerve, glial means glue. They also have diverse cells that are going to help to support and nourish these neurons. So the neuroglial cells will do that. We also will have some called microglial cells, and we'll have satellite cells that work within the nervous system. Now, looking at a nervous cell, you're going to notice some specific things there. You notice on one end, you're going to have the dendrites. That's all of these little branches coming in here. They're going to pick up on the stimuli or on the information. That information is going to travel through the cell body and then make its way to this point here where it starts to go from being a wider portion and it comes in and creates that axon. That's called the axon hillock. It's going to then travel down the axon until it's the axon terminals or those end bulbs, end plates. From there, it will connect to either another neuron or it will connect to muscle tissue. And so that's how that information will travel down. Looking over here, in this picture, we also have a type of neural cell called an astrocyte. These are going to be important cells for creating the blood-brain barrier, so they will wrap around things such as capillaries, and they'll allow different things to come across, like the nutrients, and they'll allow the oxygen to come across, and they'll take away waste products and carbon dioxide. But they do not allow anything else or not much else to cross across. So the blood can't get across, the plasma can't get across. That's going to be important because it can keep toxic substances out of the brain using these astrocytes. Now, bad thing with that is if you do have some type of cancer in the brain, a tumor, you can't have normal chemotherapy with this because it will not be able to cross that blood-brain barrier because of these astrocytes, because it keeps those toxins out. So it makes it a little bit harder to treat these types of tumors. Another cell that you see there is going to be what we refer to as a microglial cell, kind of like the neuroglial cell. It's a support cell. It's going to use phagocytosis in order to eat up things that don't belong there or old worn-out cell parts that might be within that nervous tissue. We've got our regular neuron here. You're going to see that it's got the axon that's traveling down. And remember, we talked about Schwann. These cells that you see going around this area here, those are your Schwann cells, and those cells are going to be insulating those neurons. Lastly, up here that I see, we have these ependymal cells. Ependymal cells are going to be important within the central nervous system, or the CNS. They're responsible for creating cerebral spinal fluid. And so just know that those are going to create and release the cerebral spinal fluid. They'll also have cilia on them, and so they'll act in a wave-like motion in order to move that fluid in a specific direction. All right, organs and organ systems. Organs are going to possess different types of tissues, and they're going to perform specialized roles. For example, the heart's going to pump blood. The intestines are going to move food down through the system as they start to absorb. Organ systems are going to be comprised of several diverse organs that carry out a specific life process. 11 organ systems are going to be common in most animals. Examples of these would be like the cardiovascular system of animals will consist of the heart working in concert with blood vessels to ensure transportation of gases and nutrients throughout the body. Homeostasis. So homeostasis is going to be a systemic process. Sorry, I'll move myself out the way. In which the organism's body employs physical and chemical methods to maintain a constant internal environment, despite the changes in the external environment going on. So your body's constantly going to be monitoring both inside and outside of itself, so it stays within a specific set range of whatever set it keeps. For example, the average blood pressure is 120/80, and so it's going to try to keep that set blood pressure range for you. So the idea of homeostasis is first going to be introduced by a French physiologist, Claude Bernard, in 1854. He's most influential scientist to bring the credence to Bernard's idea, including the American physiologist Lawrence J. Henderson and Walter B. Cannon. So Henderson's going to popularize the idea of milieu interior in the United States in the mid-1920s. Cannon is then going to be credited with coining the term homeostasis in 1932. So homeostasis is going to be maintained through feedback systems. We're going to have two. We have positive feedback and we have negative feedback. Starting off with negative feedback systems, this is going to help to evaluate and reset fluctuations in a physiological process, things like body temperature. This involves four main components: a set point, which your body is going to determine, that's its operational range for physiological variability. What this is basically is everybody has a specific set point for temperature in their body. Mine, I usually, when I take my temperature, it's around 98.1 or 97.9. If it gets up to what we consider to be normal is 98.6, that's actually when I start to feel bad. I feel like I have a fever, so that makes me irritable and achy. That's my body set point. Receptors, these are going to be body structures that are responsible for monitoring and detecting change within the internal environment. We also have a control center. The control center is going to be your brain. That's going to receive and integrate information acquired by the receptor to detect deviation from that desired set point that that body has, and it decides what it needs to do. Does it need to, let's say, bring the temperature down, or does it need to bring the temperature up? Then lastly, we have the effectors. These are going to be the body structures such as muscles or glands, which are going to produce a response to oppose, reverse, or reduce the effects of change. And so in a negative feedback loop, you're trying to go against what's happening. So if you're getting too hot, it wants to cool you down. If you're getting too cold, it wants to heat you up. So these are always going to be opposites. Most feedback systems are going to be negative feedback systems. Now, with a negative feedback system, these are going to be commonly used examples to understand functionality of negative feedback systems and how desired room temperature can be regulated by a thermostat linked to a heating or cooling system. And so here you see that you got a specific set point. The sensors are going to detect that temperature is rising. The thermostat is going to activate and is going to start the cooling system. So the effector would be the air conditioner. Once that air conditioner starts, it releases the AC and the room temperature is going to decrease and bring the temperature back to its homeostatic balance at its set point that it wants to be at. If the person is starting to get too cold, the sensor is going to detect that drop in temperature and the thermostat is going to activate the heating system. The effector is going to be the heating system. Usually it's going to be the muscles which are contracting, think of shivering, and that's going to actually increase the temperature of the body and take you back up to that set point. And so here you just see another visual of what's going on with body temperatures falling or body temperatures rising. Going into the opposite, the positive feedback loop. This is going to accelerate change away from the homeostasis until the triggering activity ceases. And so what this is going to do is, an example of this would be childbirth. There's not a lot of positive feedback loops. Once the contractions start and labor has started, it will increase those contractions until it expels the baby and the placenta. Once it expels the baby and the placenta, it will then start to go back and through a negative feedback loop and return it back to its normal set point. But with positive feedback, it's always going to increase what's going on until whatever process needs to be done is complete. So it's unusual in biology because if left unchecked, it can lead to instability or even death. So if you keep increasing things within the body that are going on that don't need to be increased, it could lead to death. Beneficial examples, though, that the body uses positive feedback for are going to be things like childbirth, lactation, which is making milk, and blood clotting. So those are three examples of when you would use positive feedback. Thermoregulation. So an animal's homeostatic response, move me out of the way, in maintaining a constant internal temperature is going to be basic concepts of thermoregulation. So we have our endotherms. Endotherms are going to be animals whose body temperatures remain constant from internal heat that's going to be generated from metabolism. And like I said, most of the time that comes from muscle metabolism. Birds and mammals, humans, we're all going to have endotherms. thermoregulation. So we are endotherms. Ectotherms are going to depend on heat derived from sources within their external environment. And so these will be invertebrates, these will be fish, amphibians, reptiles. These are going to be animals that like to sunbathe to bring their temperatures up. Animals' homeostatic response in maintaining constant internal temperatures. Another one is how heat's going to be exchanged between the body of the animal and the environment. So we can have conduction. That's where it's going to have a transfer of heat through direct contact between objects of different temperatures. So an example of this one would be the snake laying on the hot road in order to absorb some of that heat because it's cold. Another way is going to be radiation. This is a transfer of heat in the form of electromagnetic waves. This is going to have an exchange between objects in different temperatures that are not going to be in direct contact, so they're not actually touching each other. An example of this would be like a snake laying on a tree branch to receive the radiant heat from the sun that's coming through the atmosphere. Another type is going to be convection. So this is the transfer of heat to air or water as the medium moves over a warmer object. So this would be like an elephant cooling itself in the water. Another type, we have evaporation. This is going to be the transfer of heat during the vaporization of a liquid gas. So an example of this would be sweat on your skin. And that would be there to help cool you off. And our third type of thermal regulation, we're going to have physical, physiological, and behavioral adaptations. So blubber in marine animals is going to help to insulate them. That combined in responses of organ systems like the cardiovascular, integumentary, and skeletal systems will come together. You could have basking in the sun like reptiles, selecting a microhabitat that's going to provide shade for insects, or soaking in bodies of water like mammals will do. With osmoregulation, it's going to be able to regulate the concentration of water and ions within an animal's body. So water gained by an animal can be through direct diffusion, through osmosis, through drinking, or from water that's going to be found in the food that they eat. Body fluid concentrations are going to use things called osmoconformers, animals whose body's fluids concentration is going to be equivalent to the environment. Examples of these are going to be where most marine invertebrates have the same osmoconformers. Jellyfish, sea stars are all going to have this. So the composition of the body fluid is going to be similar or equivalent to the salinity of the seawater that they are actually soaking in. Osmoregulators in animals whose body fluid concentration is going to be different from their environment, and it's going to be maintained by specialized osmoregulators, structures that are going to be found within the body. So we had kidney-like nephedria. That's going to be found in flatworms and annelids. We're going to have gills, which are found in marine and freshwater invertebrate fish. We have malphigian tubules, which are going to be found in insects and terrestrial arthropods. And we have kidneys, which are going to be found in the vertebrates. So we can see here with osmoregulation in freshwater environments, the fish is going to be able to drink water in. It actively takes up water and ions through its gills as well, and it can absorb it or lose it through its skin. So it can excrete diluted urine, so more water than ions is actually going to be coming out, and it can move some ions, but not as much. So notice how that blue arrow is going to be a lot bigger. With the osmoregulation in saltwater environments, they're going to be able to drink ample water coming in, and it excretes water and ions through its gills. Also, it can lose and absorb ions throughout its skin, and it will excrete concentrated urine, so it excretes more ions out of its urine than it does water. All right, so this is where we're going to leave off. We will pick up with this section on our part 2 of our video.